Michael is a senior biology major from Brownsburg, Indiana, and he came to King to do nursing at exactly the wrong time. So, in light of that, he became a biology major, and we've really enjoyed having him in our major. And during his time at King, Michael has been a really key member of the men's soccer team, and he will be missed from the soccer team and also our department as he graduates this spring. And after graduation, Michael is interested in working in healthcare administration up in Indiana. So I'm really pleased to um, have him give this presentation today on neuroinflammation. So, Michael, you can go ahead and get started.
Okay, like Dr. Ong said, well, thank you for that introduction, Dr. Ong. But as Dr. Ong said, my presentation is on neuroinflammatory effects on neurodegenerative disease. The reason I got interested in neurodegenerative diseases is because my uncle was diagnosed with Alzheimer's. And when I was a younger kid, around like 9, 10, I was always confused about how it worked. And like, I was always come to my grandmother's house and she'd be the one to take care of them. And I always be confused about how every time I came to her house, like he didn't understand like who I was and like that I was related to him. And then every time he'd be like, "Oh, who's this kid again?" But then, so I was like, I need a new topic for science seminar. I'm gonna see what I can find, and then that came into my mind, and I was like, look more into the neurodegenerative diseases. So this is how I saw my topic. Now, some of these common degenerative diseases. The first one, Alzheimer's, I'm mostly gonna focus on in my presentation. There's also others like Friedreich's ataxia, Huntington's disease, Lubai's disease, and another common one, Parkinson's. Now, what is a neurodegenerative disease? Neurodegenerative disease is a range of conditions that affect neurons, and they progressively generate these neurons. And the worst part about the neurodegenerative disease is that there's no known cure. And also, there's 5.8 million people that are living with Alzheimer's dementia in the United States. Now, this picture is showing the brain activity that goes on, especially when someone has Alzheimer's compared to a normal brain, and also even mild cognitive impairment. And as you can see on a normal brain, you can see how there's a lot of more brain activity that's going on, especially on the outsides of the brain. You can see how there's red spots, you see how there's activity going on. And even in the mild cognitive impairment brain, you can still see how there's still some activity going on, especially on the outsides. And compared to the Alzheimer's brain, you can see how there's a gap in the middle. And I'm going to explain more of that in another picture later. Now, an aspect of neurodegenerative disease is dementia. This is more of a problem with the mental functioning, kind of like what goes on with the memory loss. And this diagnosed dementia is more of an impairment of at least 2 brain functions. So like, this could be like, let's say like 2 parts of your brain, like so like 2 different lobes or things like that. And also this is chronic, so it can go on for a long time or come and go away, just depending on every different case. And this is the more common aspect of neurodegenerative disease. There's another aspect called ataxia. The dementia is what more people are know about. And there's also 3 million cases of dementia in the United States. Now, this illustration is showing you more of the progression of Alzheimer's. And you can see how it goes from mild cognitive impairment to all the way to severe Alzheimer's. So first, we talk about the mild cognitive impairment. As you can see, the mild cognitive impairment is more on the medial temporal lobe, and it's more of your like, short-term memory loss. Like when someone says something to you, you may forget it because it's not that you're not really focused on it that much. And then moving into mild Alzheimer's, this is when the Alzheimer's starts to spread a little bit more. That's when it reaches more of your lateral temporal and even your parietal lobes. And this is more of like when you have problems reading or like knowing certain objects. And moving into more of the moderate Alzheimer's, you can see how this is more of your frontal lobe, which is more of your judgment, kind of your decision making. Kind of like this is what more people like start to like recognize that you have Alzheimer's. It's like that person would make a decision, like they usually wouldn't make a decision. That's what usually happens when it starts to affect your frontal lobe. And going into severe Alzheimer's, this is when it's more of like when the disease pretty much reaches most of your brain. This is more of going into the occipital lobe. This is when you start to have more visual problems. Now, the biology behind neurodegenerative disease. Most researchers and doctors believe that this is influenced by genetics from seeing, like, let's say, family trees, and you can see how how certain people are being passed down, that's Alzheimer's, and I'll show you like different like genetic factors that show why. But this is not the only reason. Like let's say like, you get this genetic factor. It's not the only way you'll get this disease. And also, it can happen through medical conditions. I have an example up here, as you can see, as a stroke, which is also an example. But there's other things that can happen, such as exposure to different chemicals or toxins on aging neurons, or even neuroinflammation, which I'm going to mostly focus on today. But even with aging neurons, let's say it's more of like a reduced function. Like not your neurons are dead, but it's more like they're not doing as much as they should be, or they're supposed to. And then also in previous medical conditions, it could also be even like a traumatic brain injury. Now going more into the treatments, like I said earlier, there's no known cure for any neurodegenerative disease, but there's also things that can help you relieve these symptoms or even increase mobility or even prolong, which most people want, prolong their life. And the way you do this is through these immunosuppressive drugs. Going to amyloid beta, this is a memory protein that helps repair and grow these neurons. And what amyloid beta does, like when it grows and repairs these neurons, also it creates these plaques that when they're clumped together, they block cell-to-cell synapses. So like such as these cells won't be able to have these cell-to-cell communication, won't be able to tell certain cells different things. And in this illustration here, you can see on the left side the normal cleavage of the amyloid beta, and you can see how it cleaves and it goes through normally. Then going on to the abnormal cleavage of the amyloid beta, this is what I was talking about more of when the plaques are getting built up. And when they get built up, they cause a little cluster here. You see this cluster here, and they get backed up, and they won't be able to go through as well. Now moving to tau proteins. These are proteins that are found in these neurons and glial cells, which are the two cells that are usually found in your central nervous system. And what they do is they modulate the stability of these axon microtubules. And what tauopathy is a class of neurodegenerative diseases that it's like its own class of like neurodegenerative diseases. And also neurofibrillary and gliofibrillary tangles are primary markers of Alzheimer's. So like these are markers that, like, if let's say getting diagnosed, you'll find these markers in people who have Alzheimer's. But these are just primary markers, so you don't have to actually have these certain markers to have Alzheimer's. And in this illustration, this is showing you how the stability of these tau proteins, how they keep the microtubules together. And then without these microtubules, you see how without these tau proteins, you can see how the microtubules get loose. And then you can see how they create these little tangle formations right here. Now moving into neuroinflammation, there's many different ways for like neuroinflammation to happen. Like such as I was saying, like it's like a pathogen can happen, or even like a brain or traumatic brain injury. These things like initiate neuroinflammation. What neuroinflammation does, what it's supposed to do, is to defend against potential harm. Such as, let's say, a pathogen is in there that it's not supposed to, they'll have this neuroinflammation go on and then try to eliminate that pathogen. But the only thing with neuroinflammation, it can be protective and destructive, depending on the intensity and the duration of the inflammation. And off this illustration, you can see, this is what I was showing you earlier from the brain activity. You can see how the inflammation in the white parts in the middle, from a mild compared to a severe. And then you can see how a mild, it's also like in the middle here, but it's not as bad comparing to like the moderate and going to severe. And severe is like more I showed you earlier in the brain activity, how most of the gap was in the middle in the brain activity. That's where the neuroinflammation is also happening. Now, neuroinflammation is more a part of the innate immune system, which is kind of like your first line of defense. Is more of a non-specific. And this is more of like your body's doing a broad like a defense system at whatever pathogen or depending on what's coming in your body. So it's like kind of your first line of defense. And so like it's kind of like an advil. You throw like it's kind of more of a broad kind of thing you try to do. But then also when this happens, it also stimulates the adaptive immune system, which is more kind of specific, such as with the antibodies or certain pathogens it's recognized before. And then when it goes through this innate immune system, it stimulates that adaptive immune system for anything that is ever recognized before. Now moving into microglia. Microglia is more of kind of another like cells that live in your central nervous system. And what they do is we move these damaged neurons from the central nervous system, and they mediate these immune response by phagocytosis. And off this picture, this is showing you a microglia, and you can see how when it gets through this neuroinflammation, it loses its dendrites. So you can see from this how it has a resting microglia. And when it gets activated, you can see how it loses its dendrites and pretty much doesn't have as well communication that's going on between the cell. And then what this microglia do are they signal receptors on for the cytokines. And then these cytokines are a group of proteins that get secreted by the cells in the immune system. And they are chemical messengers that bind to the surface of these cells. And what they do is they signal molecules that regulate this immune response. And you can see here how the cytokine producing cell is releasing these cytokines to the target cell, so it would bind to the outside, outside of this cell, and then also tell it to regulate that immune response. Now, my hypothesis: neuroinflammation promotes brain changes that lead to neurodegenerative disorders. For my first study, associations between brain inflammatory profiles and human neuropathology are altered based on apolipoprotein E epsilon 4 genotype. Why this study was done? This study was done to test the hypothesis that microglia and Alzheimer's implicated cytokines were differentially associated with Alzheimer's pathology based on the presence of APOE epsilon 4. Apolipoprotein, also known as APOE epsilon 4, is a fat-binding protein which carries cholesterol and other fats and even other proteins into the bloodstream. This is a pro-inflammatory, and this is what I was talking about earlier, how I was talking about the genetic variant, on chromosome 19. This is what I was talking about earlier, like as I said, how it's more, how doctors and researchers believe how it's more on the influence of genetics. But this is a common genetic risk factor in dementia. So let's say you have the genetic variant, but this doesn't mean that you actually are going to get Alzheimer's or dementia, but it's giving you a higher risk of getting Alzheimer's or any type of dementia. Now moving to the methods. There was 154 autopsy patients from the Framingham Heart Study, 119 APOE epsilon 4 negative, and 35 APOE epsilon 4 positive. And what they did was analyze the brain tissue for changes that happen in the microglia and the cytokine levels. Table 1. This is showing you the sample size compared from the APOE epsilon 4 negative and the APOE epsilon 4 positive. And then it's also showing you the age of what these people died at. Like I was telling you, it's more of older people, like aging neurons, like more of a reduced function. So you can see how people who have APOE epsilon 4 negative are 87.9, and also APOE epsilon 4 positive were 85.9 average age. And moving into we'll see if it's more common in females or males. You see how the percentage of males were 43.7% APOE epsilon 4 negative and 45.7% APOE epsilon 4 positive. And showing you the more important one on this table is more of the dementia. And you can see how in the APOE epsilon 4 negative was 46.2%, and then the APOE epsilon 4 positive was 65.7%. And you can see how in the p-value how it's below 0.05, showing you that this was statistically significant. Now, tauopathy versus microglia levels. IBA1 is a receptor on these microglia cells, and this is showing you. You can see how all the blue circles are APOE epsilon 4 negative patients compared to the APOE epsilon 4 positive patients that are in the red squares. That's what's showing you how you can tell how there's a lot more blue circles compared to the red squares. There's a lot more APOE epsilon 4 negative patients. And on this graph, you can see how in the APOE epsilon 4 positive trend line here, you can see how there's a more positive correlation comparing the IBA1 to tau pathology, showing you there's a better correlation compared to the APOE 4 negative. You can see how the correlation is more a straight line compared to an upward slope line. showing you that there's a more correlation between tau pathology and IBA1 in APOE epsilon 4 positive patients. And moving to table 2, this is showing you the cytokines that get altered when it comes to the APOE epsilon 4 positive and negative patients. And I want you to focus more on the IL-13 and the IL-4. And you can see how the APOE epsilon 4 negative was 4.02 in the IL-13 compared to the APOE epsilon 4 positive patients. And you can see how this is significant. And also, even in the IL-4, the APOE epsilon 4 negative patients were 0.302 compared to the APOE epsilon 4 positive patients that was 0.2601. And also, again, it's also statistically significant. And this illustration is showing you on the left side, the APOE epsilon 4 negative patients, how they don't produce these certain cytokines. Oh, they do produce these certain cytokines that block you from these neurofilament tangles from getting tangled. Like compared to on the right side, the APOE epsilon 4 positive patients, they don't produce these cytokines. So they're allowing these neurofibrillary tangles to get the blockage from the tau proteins, which I said earlier. The takeaway point from this first study, there's a positive correlation between microglia and tauopathy patients, which is higher in the APOE epsilon 4 positive patients. And in the APOE epsilon 4 positive patients, they lose their ability to inhibit these neurofibrillary tangles compared to the APOE epsilon 4 negative patients. Now moving into study 2, systemic inflammation impairs microglial alpha beta clearance through NLRP3 inflammasome. Now why this study was done. This study was done to examine the changes in the microglial cells during inflammation caused by induced bacterial LPS. LPS is a lipopolysaccharide, which is an endotoxin that is on the outer membrane of this gram-negative bacteria. And this, what it does is stimulate the release of these cytokines. And also, it creates a small inflammatory response for these pathogens that may come into your brain. Like I said earlier, with the innate immune system does. Now, showing you what LPS is. LPS is here. You can see the circle here. You can see how LPS is on the outside of this gram-negative bacteria. Like I said earlier, it's on the outside, the outer membrane of the gram-negative bacteria. You can see it goes all the way along here. Now the methods for this second study. They use a cranial window surgery on mice, and what they do is inject these with a single dose of LPS. Then these glial cells that are in the central nervous system are analyzed at day 2 and also day 10 after injection of the LPS. And how they do this is how they measure these glial cells are using a 2-photon laser scanning microscopy. Now this is the illustration of kind of what they did to the mice. Like it's showing you at base when they inject the mice with this LPS, and also at day 2 when they inject the mice with LPS, and also at day 10 when they inject these mice with LPS. Now this is showing you the branch number, the path length, and also the branch order. To define these three things, I'm gonna show you here. So first, with the branch number is more of the number of branches. See here, like you can see how it gets less and less. It's more of the number of branches that are coming from these glial cells. The path length is more of the length of these branches. You can see how it goes from, like, you can see how there's a lot of branches there, how long they are, and comparing to like how short these are in the 48 hours, not as long as here. And the third one is the branch order, which is the most complex to describe pretty much. But the branch order is more of kind of showing you how there's a lot of branches that come off of a different branch. So like on 48 hours, you see how there's one long branch by itself, but then you see how there's more on this right side. You can see how it breaks off into 2 different branches. And you see how it happens more often at the 24 hours or even more at the 8 and 4 and base. And that's kind of describing how the branch number, the path length, and also the branch order. Now moving into, like, this is more of the graph of what's showing you what's happening. So you can see how the number of branches go down from the base to the 48 hours. And you can see how it goes from like at least 70 and down to almost at 30 in those 2 days. And then you can see how the graph is showing you the stars, how it it's showing you the statistical significance of this. It's showing you from the base until 24 hours is 1-star significance, and moving on from the base to 48 hours is a 3-star statistical significance. That's the number of branches. Now moving into the path length, which I said earlier is more the length of these branches, is showing you kind of the same trend from 400 to also down to 200 in those 48 hours. But then it's also showing you the statistical significance from the 2 stars within the 24 hours, and then the 3 stars statistical significance for from the whole 48 hours. Then moving into the branch order, this is kind of, once again, showing you kind of the same trend that's going on. And then you can see how there's a 1-star significance going on for about 8 hours, and then going on to a 3-star statistical significance from the base to 24 hours, and also again to 48 hours. Now moving to a second part of the method, like on a second part experiment of this second study, it's showing you of more of a green fluorescent glial cells. And what the difference is, these mice have NLRP3 knockout mice, which means they don't have this NLRP3. They can't produce these inflammasomes. When NLRP3 is a NOD-like receptor protein 3. Protein 3 is the actual name of it. And what they do is they regulate inflammation. The way they do this is they initiate apoptosis and trigger the release of these cytokines. Now this is what NLRP3 is right here. And then you can see how it has pathogen activators. And then from the pathogen activators, they are bacteria-derived, virus-derived, fungus-derived, and also protozoa-derived. There's also even sterile activators, which come from environment-derived and also even self-derived. And in the self-derived, you can see how there's amyloid beta here. And once again, this is showing you the mice again, for how they use the cranial window once again. And then they remove the NLRP3 from these mice, which is called NLRP3 knockout mice. And you can see how they have the same cranial window from the base to 10 days. And you can see off on the left side the ages of these mice. So like these are 5 months old mice on the left side compared to the 15-month-old mice on the right side. And then the wild type are the mice who do have the NLRP3 compared to the mice who do not have the NLRP3. And then you can see how in the wild type, in the first 5 months wild type, you can see how the glial cells look normal compared to the day 2. The day 2, you can see how some of the glial cells are missing a little bit. You only see like around 3 around here. And you can see how there's a lot more missing compared to what's going on in the base. And the day 10, you can see how a lot more of the glial cells came back. And also in NLRP3, you don't see this difference. You kind of see more of the same glial cells from the base all the way to 10 days. And then moving into the 15-month mice, you kind of see the same trend going on. So you can see the same trend going on. You can see how the wild type mice from the base have a lot of glial cells. Moving into day 2, you see how a lot of the glial cells kind of go away. And moving to day 10, a lot of the glial cells even come back. And in the NLRP3 in the 15-month mice, you kind of see the same trend as that 5-month mice. You kind of see a lot of glial cells in base. Even in the day 2, you still see a lot of glial cells. And then even in day 10, you still see kind of the same amount of glial cells. Now, moving into the number of, kind of the graph we're showing you with the number of branches. For the wild type 5-month mice, which is here, you kind of see the same trend which was going on in before. It's kind of the same thing from the base to 2 days. You see the kind of a drop that's going on, but you still see how the statistical significance in it with the 2 stars. But then in the 10 days, you see how it comes back up almost around kind of almost even. And then even in the 5 months with NLRP3, you kind of see how the 2 days don't drop as much. And it's showing you how the NS, which means no significance, is showing you how there's no significance from the base to the 10 days. Then moving into the 15-month-old mice, and the reason I believe they have less number of branches compared to the 5 months is because I feel like these mice are a lot older, so they don't have as healthy or as like they have these reducing neurons, reducing the function of neurons. Like I said earlier, the aging neurons. So I think really they have a number a lot less number to start with to begin with, but kind of shows you the same trend, but just in the older mice. But then you can see how from the base to 2 days to 10 days, you can see how it went from down to 2 days and back up to 10 days. And you still see the statistical significance. You can see how there's a 1 star in between the base 2 days and even the 2 days to 10 days. And then the NLRP3 mice that are 15 months old, you kind of see how there's no significance, kind of the same thing, kind of like it's more of a steady amount of branches that are in these glial cells. Now moving into the path length, you kind of see the pretty much the same thing that was going on, and even in the branch number, you see from the base to 2 days, you can see how there's a drop off, and moving back into the 10 days, it comes back up, and you see how there's a two-star statistical significance going on here. And moving into the NLRP3, there's no significance once again, kind of showing you like there's a path length that goes down a little bit after 2 days, but it also comes back up after 10 days. And moving into the 5 months wild type mice, you see kind of the same trend once again as the branch number, a lot less glial cells, but kind of the same thing in the path length. From base to 2 days, you still see the statistical significance all the way back up in the 10 days. But then moving into the NLRP3 mice, you kind of see how a lot of the mice, the knockout mice, had from the base to 2 days don't go down as much, but then even to 10 days it comes up. But it still shows you the NS showing you that there's no significance in these. And then also in the maximum branch order, you kind of see how pretty much the same trend once again going on from the wild type 5 months, NLRP3 5 months, the wild type 15 months, kind of the same thing what's been going on in the path length, and then the branch number, and then again also in NLRP3 in 15 months. Now the takeaway points from the second study. Glial cells, they lose more branches with time, but when they reach these 10 days, some of these glial cells branch orders, branch number, and the path length, they start to regenerate after the next 8 days, after the 2 days that happen. And also, the absence of NLRP3 allowed for these branches of glial cells to have a lot longer branches and a higher number of them. Now, my restated hypothesis. Neuroinflammation promotes brain changes that lead to these neurodegenerative disorders. Now, with the future studies, I feel like there should be a bigger sample size in the first study, because I feel like in the first study, there's 119 APOE epsilon 4 negative patients compared to the APOE epsilon 4 positive patients. There's only 35 of them. I feel like a bigger sample size would show us maybe a bigger difference that's going on in those going on with the cytokine levels compared to tau pathology levels. So I feel like a higher number of at least APOE epsilon 4 positive patients would help the study a lot more. And also for the second study, most of this experiment, the whole experiment was done on mice, but also would it work the same for humans? Because obviously we're different than them. So would we also have the same, let's say the same like branch number, branch order, and a path length of these glial cells? Would it work the same for us without the NLRP3? And then sources and any questions.
Hey, Michael, when you mentioned about inflammation being both protective and destructive, I mean, the destructive part is pretty obvious for us. What do you mean by it being possibly protective?
I would say that neuroinflammation, like there's a reason we have it. It's part of our innate immune system. I would say it's protective because let's say a pathogen comes into our body. Our body's first action would be through the innate immune system, which would be the neuroinflammation, trying to heat our body up, heat our brain up, like our central nervous system, trying to get rid of that pathogen, trying to make a tough environment for that pathogen, so we can get rid of those certain pathogens that can't live through that. And then eventually, but the thing is, like I was saying earlier, it has also the destructive effects that I was saying, which also hurts us.
So other than raising the heat, inflammation, does that serve any other purpose in getting rid of the pathogen that's coming in its protective mode?
You said other than heating up, what else does it do? Is that what you're asking?
Other than raising the temperature in the quest of ridding the pathogen, what else goes on in inflammation other than the raising of the temperature? Do you know?
I don't know for sure, but I know like it's more of a protective for like, I know it's like more protective for like I said, for those pathogens. But also it was saying how it's protective for, let's say, like those brain traumatic injuries. Like I was saying earlier, like whenever those things happen, and like I guess like our brain like kind of like shuts down for a little bit, and so we'll be able to like recover those neurons and those certain glial cells, so we don't lose them. Like they don't, like, so we don't lose like, so we only get born with a certain amount of neurons, and we don't lose those like through those traumatic brain injuries.
Okay.
Hey, Michael, this is Dr. Vaughn. You showed a lot of graphs for the second paper in particular that had stars on it. You mentioned the difference. This one has 1, 2, and 3. Why are those relevant? What does 1 star versus 3 stars mean?
Like, so like 1 star would be like, I think more of a higher p-value compared to 3 stars. That would be like, what that means more, it's more statistically significant compared to the 1 star.
Okay, thank you. And then second quick question is, you said something about mice knockout. What is a knockout mice? What does that mean?
Like the knockout mice is pretty much the mice that like, it's kind of like they knock out the NLRP3, so they have that, like they don't have that an NLRP3 in those mice. So that's kind of like showing you how they don't go through that same neural inflammation, so they don't have that neural inflammation in their brain that goes that kind of like decreases their branch order, decreases that pathway.
Okay, thank you.
Any other questions? Okay, thank you, Michael. And for the instructors, remember to send your scores and comments to me by noon tomorrow. Next week, we will have 2 more presentations on Monday. I'm not sure yet if they're going to be completely Zoom or with some of us in the room, but I will let you know that before Friday. So thank you, everyone, and have a great rest of your day.
Can I make a
Thank you, guys.
real quick before
Michael.
the MFAT exam? If you're a biology student and you're graduating this semester, you've got to take the MFAT. So please
Okay, yeah.
schedule. It's not just for you, Michael. It's for anyone out there that's listening to this, because not everybody's scheduled it yet, and some people aren't responding to me about it. So
Okay, all right, thank you.
yeah.
Thank you, Michael. I enjoyed your presentation.
All right, thank you.